Hello, in this video we'll talk about the principles behind acid-fast staining. Acid-fast staining is a differential staining method used in microbiology. The stain is used to identify acid-fast organisms such as that of in the genus Mycobacterium. In this staining, the acid-fast bacteria would appear reddish, whereas the non-acid-fast bacteria would appear blue. In this video, it would be totally clear what is the basis of this differential coloring. But let's understand what are the acid-fast organisms. Acid-fast organisms are characterized by the presence of wax-like nearly impermeable cell wall. They have very high level of mycolic acid, large amount of fatty acids, waxes and complex lipid which make their cell wall literally impermeable to all the substances. So their cell wall is so much resistant that they don't take up any other stain such as Gram stain. And acid-fast organisms are highly resistant to disinfectants or dry conditions. And we need to do this kind of acid-fast staining to identify Mycobacterium tuberculosis. And that's why this is really important from a clinical perspective as well. So let's hit the virtual lab and try to do a acid-fast staining. First of all, let's get introduced with the reagents. We need carbol-fuchsin as the primary stain, then methylene blue as the counter stain. There would be acid alcohol, which would be the decolorizing agent. There would be distilled water for washing purposes, and some other reagents like pipette, forceps. All of these things would be very common to any microbiome lab. So first step involves smearing the sample on a slide. Here the patient-derived sample would be smeared on a particular slide, and we are expecting that this particular sample might have Mycobacterium tuberculosis in that. At the end of this video, we would understand whether it has it or not. So the primary stain would involve carbol-fuchsin. Carbol-fuchsin is actually a particular stain which has phenol in it, and it can really penetrate even this impermeable mycolic acid containing cell wall. So they would penetrate and get inside the cell wall. Carbol-fuchsin actually has basic fuchsin, ethanol, then phenol, I mean the heat melted crystals of phenol, and also distilled water. So all the bacteria at this particular step would be colored red with the carbol-fuchsin, be it acid fast or be it non-acid fast, and this incubation would happen for, let's say, 30 seconds. After this step, there is a heat fixation step which would fix the stain even further, and it's kind of important such that these bacteria should adhere to the slide and should not wash away when we are adding other reagents. Now in the step 3, we would be using a decolorization agent. After applying this decolorization agent, the bacteria which are non-acid fast cannot retain this carbol-fuchsin stain. This decolorizing agent is very strong, and this decolorizing agent contains ethanol and hydrochloric acid. So it would wipe out all the staining from the non-acid fast organisms. Remember, the acid fast bacteria has very rigid impermeable cell wall. So actually this decolorizing agent cannot wipe out the stain from these acid fast bacteria. So by the end of this step, the acid fast bacteria should retain their red staining, whereas the non-acid fast bacteria would fail to retain their carbol-fuchsin stain. Now in the last step, we would be doing counter staining with a counter stain popularly used known as methylene blue. It's a blue colored counter stain. So obviously the bacteria which has failed to retain carbol-fuchsin now would take the counter stain methylene blue. Remember, these acid fast bacteria has very impermeable cell wall, so they would not allow methylene blue to stain them. So acid fast bacteria would retain their red coloration due to carbol-fuchsin, and the non-acid fast bacteria would ultimately take the counter stain methylene blue, and thereby they would be visualized as blue cells. So there are two methods used in the microbiology lab. One is Zeil-Nielsen method and another is Kinyoun method. Both these methods are useful for microbiologists. And you can take a look at the reagents really quickly, but I'm not going to get into details of these exact protocols. Now, as per summary, we have used a primary stain carbolufusin, which is highly permeable. It can even penetrate the almost impermeable cell walls of an acid-fast bacterium. Now, after that, we have used a strong decolorizing agent such as acid alcohol, which would wipe out all the stain from non-acid fast bacteria, and we have counter stain with methylene blue, where the non-acid fast bacteria took a blue stain and the acid fast bacteria has retained their red carbolufusin stain. This video tells us what is the basis of acid fast staining and how it is really important for medical microbiology.